Hello everyone, and a warm welcome to today's webinar. We are excited to explore the topic cell therapy in autoimmune diseases from clinical results to patient access. Thank you for joining us. Our speakers today are Cristina Conforti Andreoni, Marketing Manager for Portfolio Strategy in Immunotherapy at Miltenyi Biotec, Dr. Tobias Kappenkerl, Vice President for Early Clinical Research and Development and Translational Medicine at Miltenyi Biomedicine, and Dr. Dick de Vries, Director Clinical Research and Development at Miltenyi Biomedicine. Before we dive in, let me briefly walk you through the topics so you know what we'll be covering today and what you can expect to take away from this session. We'll begin with an introduction to cellular therapies in autoimmune diseases, focusing on their mechanisms of action. Next, we'll see examples of clinical outcomes of CAR T-cell therapies in autoimmune diseases. And from there, we'll explore key considerations around clinical trial design and patient monitoring for CAR T-cell therapies. We'll then shift to the challenges and opportunities in manufacturing of cellular therapies. And finally, we'll wrap up by discussing current and future access to cell therapies for autoimmune diseases. By the end of this session, our goal is for you to have a clear understanding of the science, the clinical landscape, the operational challenges, and the access considerations around cellular therapies in autoimmune diseases. Please now take a moment to review the disclaimer displayed on the screen. This disclaimer refers to our Miltenyi Biotec products and services. All information, interpretations, views, and opinions contained in this presentation are based on a careful review and professional judgment of the sources of information cited by the presenter. The Miltenyi Group is not liable for any interpretations, views, or opinions expressed in this presentation. And with that, let's get started with Cristina Conforti Andreoni, who will take us through the mechanism of action of cellular therapies in autoimmune diseases. Cristina, over to you.
Thank you, Georgia, and welcome everyone to our webinar. As mentioned, our topic today is cell therapy in autoimmune diseases. So I would like to start giving an overview on the current approaches to treat autoimmune diseases in the standard of care, but also as experimental therapies. As you all know, autoimmune diseases is due to a reaction of the immune system against the self, against the body. So one of the oldest and most established treatment for autoimmunity and standard of care is immunosuppression. So cooling down the system using corticosteroids, for example, but also next generation treatments and therapies like small molecules that target molecular mechanism like inflammatory pathways like JAK and BTK inhibitors. A more sophisticated approach is the one of immune retuning, and this has been established already as experimental therapy since some years. And the immune retuning is a very smart approach because try to a bit hijack the current immune system of the body of the patients by inducing a stronger regulation and tolerogenic effect using the cells that naturally already are tolerogenic. For example, the regulatory T cells, mesenchymal stem cell, dendritic cells, NKT, and other cell types. Another more drastic and also very efficacious approach is the so-called immune resetting. And in the presentation today, we are going to focus on this approach. Immune resetting can be done with autologous transplantation. So the logic is to remove all the immune cells that could be pathogenic. But of course, with the transplantation, you basically remove every cell of the hematological compartment. And then the beauty of immune resetting is that when the cells come back, they are not pathogenic anymore. So you're kind of like switching off and on the system, and that reestablish then in a non-pathological way. In a more sophisticated way to do immune resetting is using depletion of B cells specifically. This can be done with the monoclonal antibodies, or exactly as we're going to discuss today with CAR T cells and CAR NK. And another way to deplete specifically B cells or eventually T cells, it would be to use specific antibodies. So for the purpose of this webinar today, we will look into the CAR T cell therapies. So to start, we know that this audience is mixed of people that might be or not familiar with CAR. So let's give an introduction, a very short overview, what is a CAR. So as the name says, it's a chimeric antigen receptor, and chimeric comes from the Greek word chimera, which basically means a mythological animal that is a hybrid composed of two different animals. And this is exactly the purpose of the CAR, to be the best of both. So it takes the best part of a B-cell antibody, which is the specificity, and then it takes the best part of a T-cell receptor, which is the intracellular signaling that allows the cell to be a killer. So the CAR is very artificial, but very effective, recognize surface proteins, so it doesn't need a presentation of an antigen, and then immediately transmit the signal inside the cell and drive to the killing. So a CAR T is an engineered T cells that has been engineered to present on the surface a CAR, and therefore, depending on the CAR specificity, recognize target antigens on target cells, and via this internal signaling then kills the cell. So this approach can be used for B-cell malignancy and has been successfully used for therapy against B-cell cancer, especially blood cancer. But it's also recently has been applied for autoimmune diseases, because B-cell are also the pathological driver of many autoimmune disorders. The current status is that all these therapies are still experimental, but there have been good and promising data showing results in rheumatic diseases such as lupus and others, neurological diseases like myasthenia gravis, multiple sclerosis, and also more recently hematological diseases driven by autoantibodies and even gastroenterological autoimmune disorders like ulcerative colitis. So now my colleague Tobias will go in the detail of which are some examples of good clinical outcomes from CAR T-cell therapies in experimental approaches. And he will go a bit more in the detail also of how these therapies can be designed, and also which are the possibilities also for the future. Thank you, Tobias. Over to you.
For many thanks, Christina. Good morning, good afternoon, good night, everyone. I think we have a global audience, which is great. Thank you for having me here. Before I launch into the slides, just stay on the intro slide for a second, please. I want to say what this can be. I mean, here's a global development ongoing of CAR Ts and autoimmune diseases, and we are not able to give you a holistic review of the data currently. So that is not possible. But the idea is to transport the paradigm with some example data that is sort of reflective of the general evolution of what is going on. And also, I do want to note this is all about drugs in clinical development. This is early clinical data, so this is all important to note whenever you talk about this. This is not approved drugs. This is all clinical development. Another thing I want to say up front is there's a history about this, and I know we have a mixed audience. I guess there's a couple of physicians in here, but also other healthcare professionals, broadly speaking. And I think it's very interesting, since I joined this field, to understand where things are coming from. I myself am a hematologist oncologist as a background, which is good for the CAR T, which is partly difficult for the autoimmune diseases, but I'm learning a lot. The rheumatologists in the room here will realize I'm not an expert as you are probably, because it's very complex diseases. However, everything started in 2021 when the first patient ever received a CAR T product with autoimmune disease. It was a young lady treated in a German university hospital in Erlangen with a CAR, and she had a very advanced lupus disease with lupus nephritis, exhausted all available therapies. And in the German framework, there is a named patient approach for very desperate cases. And under those conditions, this young lady and their treating physicians made a bold decision to attempt CAR T treatment. And the idea was to introduce a genuine B-cell reset, which Christina already alluded to, and I will show you on a slide. Now, what happened then, since 2021, this young lady went into, and this has been published a couple of times, went into a complete remission of her disease up until today. So that's really fascinating. She was in a desperate state, exhausted all available treatments. This was the first attempt ever of this approach, and she is now a young lady studying. I was lucky to talk to her recently. She's doing fine, and she considers herself no longer being a patient. So this is what started the whole evolution. And then after those patients got treated on a single individual basis, studies were introduced. And I will talk you through one study briefly that is, I think, a foundational starting point of other studies in the field. Now it's a global field with many players, many constructs, and a variety of disease types investigated. That's important to note. So now, having said this, let's please launch into the first slide, which gives an idea of what Christina just explained verbally. Here's a little graphical illustration. So B-cell targeting therapies are established in autoimmune diseases, and they do work. And the idea is, of course, you reduce B cells, you what did you say? You silence down the immune system generally. So, however, what was not achieved so far with B cell targeting drugs is long-lasting remissions without continued treatment. And the idea why CAR Ts may be able to provide this, you can see on the right-hand side. Quickly, the purple cells are the autoimmune cells, if you like. And with monoclonals, you can't introduce a genuine B-cell reset. You don't get rid of your autoimmune population completely, so you have a repopulation of your autoimmune driving cells. Whereas the CAR T is able, at least in this graphical illustration, to eradicate all your B cells, including the autoimmune ones. It spares out, at least in the CD19 targeting example here, your plasmablasts, which is great because you keep your long-term immunity, vaccination, for example, which is good for fighting infections, of course. And then after you eradicated all your B cells, you repopulate with your normal B cells without autoimmunity. Very oversimplified, but it's not only wishful thinking. If you look at the left-hand side, histopathology, this is lymph nodes from patients treated with a monoclonal rituximab. That's the where you can see that post-treatment, you still have CD19 positive, CD20 positive B cells in your lymph nodes, whereas the middle row is post-CAR treatment, where all B cells, inclusive of the tissue, got eradicated. And that's the idea, right? This allows for the B cell reset. The CAR can go everywhere and kill all B cells around and lead to this genuine reset. And then now, here's a very nice illustration from Georg Schett from Erlangen, who is leading, I think, in many regards, this evolution. And it's a sketch of what this could look like. The current traditional scenario is you have disease activity, which is high initially. You install treatment in autoimmune diseases. You need continued treatment to suppress activity. You will have flares up and down of disease activity. Usually you need continued treatment or reinstallation of treatment. And this is the course of a patient, very generally speaking, with a burden of treatment, the burden of disease. Now, in this visionary scenario, you have a one-time treatment, CAR T, which leads to a resolution of your disease, and then over many years to come, hopefully, you stay without the disease. And I want to say it says visionary scenario. That's true. Again, we talk about early data. However, in data sets, you can, in some patients and disease types, start counting in years now. So it's not that you only know about the very initial part. You can talk about long-lasting remissions in those patients. So now here, let's dive into one study. And I told you this is one example. And I think it's a foundational one because this is one of the earliest studies that were set up in 23 by the same group that did also introduce this paradigm into autoimmunity. This drug is now called Zorpocel. This early phase 1/2 study investigated 3 different disease types in those early days: systemic lupus erythematosus, dermatomyositis, and systemic sclerosis, all complex diseases by itself. And you see in the information below, there is a variety of efficacy endpoints for the part 2, which partly involve complex scales and measurements. We don't have the time to go in there, and I'm happy to not have to do this because it's very complicated. But in the end, here's a safety part, and there's a second part, which also looks into some efficacy. And now I want to take you through like a glimpse into those results. And we should always start in early clinical data, of course, with safety. And many of you will know that CAR T cells are, of course, well established in oncology. There's a variety of CAR T products approved for lymphatic malignancies and also for multiple myeloma. And there's a distinct safety profile established of those CAR-Ts. And very importantly, there is one side effect called cytokine release syndrome, CRS, which is like an overshooting inflammatory reaction early in the treatment with a CAR-T. And what was observed here in those 24 patients, you can see by columns, there's 10 SLE patients, 9 scleroderma patients, and 5 myositis patients, that the profile was pretty benign overall. The CRS was maximum grade 2 in 2 patients and was predominantly grade 1. And to translate this for the ones who are not so familiar with this grading, CRS grade 1 is more or less a fever. So it's quite manageable. Sometimes you will install also a preventive treatment with tocilizumab in this case of a CRS grade 1. Sometimes it could only be some paracetamol. But it's a very manageable event in this population. There's another very important finding of CAR-Ts, which is a little bit less frequent than CRS, but can be also very severe and important. ICANS, that is a central nervous occurrence of side effects leading to confusion, paresis, and many other neurological findings. This luckily has not been observed in this population of autoimmune patients in a single instance. So now there's another important aspect, which is cytopenias and infections, also a known area of interest in CAR-T treatment from oncology and in focus all the same for autoimmune patients. And the group of Müller et al. Did investigate the difference of the safety profile of autoimmune patients treated with a CAR-T versus patients treated with lymphoma with a CAR-T. So to better establish now what is the difference in the side effect profile. And to start with the profile of cytopenias, you see platelets, hemoglobin, and neutrophils. And most importantly, for the ones I think most important, neutrophils and platelets, you see the red-orange line is autoimmune patients versus, sorry, it's the other way around. The blue line is autoimmune patients, and the red-orange, whatever that is the lymphoma patients. And you can see that the counts are much higher in your autoimmune patients, so you have less findings of cytopenias, and also on the slide is at the bottom, you have less lower frequencies of longer duration of neutropenias or thrombocytopenias. So overall, as for CRS and ICANS, the profile of CAR-Ts in autoimmune disease appears to be better manageable and less severe as in oncology based on this early clinical data we have. Another piece you can see on this slide is your vaccination titers. They don't disappear following CAR-T. This goes back to that CD19 targeting spares out the plasmablasts in some parts, so you keep your long-term immunity. So now let's look a little bit at efficacy. And to me, I mean, those pictures say more than 1,000 words, of course. This is, again, the same study. This is zooming in on the lupus erythematosus patients, the 10 in the study. And just let's take the picture as an example. You see this young lady being heavily affected with a skin rash. And you can also see the moon phase from long-term high-dose steroid needs she must have had. And then 4 weeks into the treatment, it's incredible to see. She really, from the visual appearance, I have never spoken with her, of course. She looks like a normal lady again, and that's just incredible. And if you look at the outcomes of the scales and the efficacy endpoints, you see everything trending down. DORIS, that's a key outpoint in lupus, which is more or less something like you don't have any disease activity measurably left. Is achieved in almost all of those patients. And you can also see on the right-hand side the disappearance of the autoantibodies over time. Zooming into another disease type, which is the next one to come, I think, is systemic sclerosis. Whoever has seen this disease in your clinical career, I think it's very impressive to see those patients. At least the ones I've seen as an oncologist who underwent like autologous transplant, I found deeply impressive. Limiting often is the lung function over time and often has a fatal prognosis in rapidly advancing patients. And here, those patients, importantly, you see the lung function measurements like forced vital capacity and the DLCO. Things were either stabilized or improved. And that's most important, right? And also you see the skin thickness was trending down, as well as those complex composite scores all really trended towards resolution or improvements. Noteworthy is that in most disease types, damage to tissue which is not inflammatory in nature. I mean, this is not a miracle drug. You can't revert back damage of tissue. Like if there is sclerosis, a CAR T won't remove sclerosis, but it will remove or can remove at least inflammation. So that's a component, and then stop progression. But you can't revert back such a severe change in tissue to normal again. And I think the last missing disease entity in this study is the inflammatory myositis. And the pattern repeats simply with other measures here. So MMT, the scores you see on top, that's your muscular strength tested with some physical activities. This is trending up. The most impressive to me, not being too familiar with all the scales and clinical daily practice, I have to admit, but the CK as a marker of the ongoing damage in your muscles, and like in the yeah, exactly there, is like trending down and normalizing in most of the patients over time as to me a very strong marker that inflammation got removed from the muscles in those instances. Lung function also plays a huge role in myositis. And you see for the affected patients here also stabilization or improvement. So that was a rapid go through over some pieces of clinical data in those 3 example disease types from the foundational CAR T study using Zorfocell, a CD19 targeting CAR. And with that, I would like to hand it over to Dirk de Vries, who will give you some more insights into clinical trials and considerations around those for autoimmune patients with CAR Ts. Dirk.
Yeah, good morning, and thank you, Tobias, for this overview of the data that has been generated so far. I apologize upfront for my humid voice and still recovering from being not hearable at all. So I'm happy to be here with you today. As Tobias showed, some high-level results from studies, from the main study we performed initially. Most of you will have seen this kind of data being presented in congresses or in journals that you read, maybe also as background to guidelines in general, if you look at how study data are used in clinical or to guide clinical practice. What I will do, as Tobias already mentioned, is doing a step back and how we create studies that ultimately result in this valuable information that's driving the progression of medicinal treatments in clinical practice. And I will do that keeping in mind your potential participation in studies or when you want to design a study yourself. What I will not do is going into consultations with health authorities, advisory boards, patient organizations, such as that's mostly not where in the interactions with companies like ours you will be involved in, and so it's a bit out of scope of how we design the studies. Another thing I want to mention up front is, as you will know, there are basically 3 phases in clinical development. They all have their own typical design requirements, but the basic principles I will present are applicable to all, and the perspective I mostly will use in this presentation is the late phase, the pivotal registration studies, but as said, the principles are basically the same. In this first slide, what you see here is the objectives and the endpoints. It all starts with creating a plan. Let's call it a wish list. What is the product profile to be? Then we create a clinical development plan. What studies will contribute what of those objectives to the plan? In the submission documentation at the end of the process, when we go for marketing approval, you have to cover safety and efficacy. That's pretty obvious, and that's also what is mostly discussed between patients and physicians in the consultation room or at the bedside. But also of value is knowledge about the pharmacology. What does the body do with the drug and how it's metabolized, and what other effects do the compound treatment has on the body. Mechanistic insights are valuable to support that information. And some studies also collect patient experiences or health technology assessments, so information about the socioeconomic impact of the new treatment. We will not, like Tobias already mentioned, go into the details of all the abbreviations mentioned here, and those are coming from lupus, so rheumatologists will recognize some of those, but the main message here is that we carefully have to select the most appropriate endpoint, as statisticians call those measurements, and the timing is also very critical, as immune systems may develop their response effect over time. So a patient may, in the early phase after administration, not respond similar as they will respond later after, for instance, a year. And there are publications already showing that renal function is, for instance, improving up to 1.5 years after administration of a CAR T. Even that is to be kept in mind with designing the study and how those endpoints relate to it. And what you see here is in the upper line, safety measurements, then also efficacy endpoints, patient-reported outcomes, and the pharmacology and biomarker assessments are included in the overall plan of a study design. Your study has to follow regulations. That's applicable to us, but that's applicable to everyone. And health authorities have their opinion about choice of endpoint, the population requirements, and also about, if available already, like in lupus and in most rheumatology, or in the major rheumatology indications, there are treatments available already. Then there are requirements to compare your new treatment to the already established effect, so that has impact on the design of your study. And most of the studies are therefore designed as a comparator study. Only if you can prove that your compound is really curing the disease, then a single arm study can be designed. And this all also has specific requirements for the population. Only for the population you choose in your trial, you can get an indication on the market. So do you, in the case of lupus, select the broader systemic lupus population, or do you limit yourself to lupus nephritis? Or even cutaneous lupus is now becoming a specific area of attention also with the authorities. And in general, as mentioned already, there are about 80 diseases in autoimmune driven by B cells that are potentially drug or disease targets for CAR T treatment. There are also guidances from EULAR and ACR, so the international societies of rheumatologists, that guide you on how to select the patients. They are also available for lupus nephritis. To set the disease activity in a study, there are several measurements. Here is mentioned the BioLec, and the 1A and 2Bs indicate only the severity of the disease. You can also apply, for instance, the SLEDAI, which is another disease activity measurement, and a lot of other criteria like age, and also exclusion criteria to limit the population and to limit the patient's risks in the study needs to be included. And this is all be consistent with each other. Choice of endpoint versus population. Lupus nephritis has a different endpoint than systemic lupus. There are all considerations, and external advice is welcome to that, consultation of the health authorities as well. And with this, it becomes a bit more operational. It's always good to visualize the design of your study. And let me walk you through this slide. On the left hand, it starts always an informed consent. We have to tell the patients what we will do in the study, what the treatment is about, what the potential risks are, and then the patient can consider whether he or she wants to participate in the study. Once agreed, there's a window to screen the patient and to see if the patient fulfills the eligibility criteria that has been set up for the study with the guidance of the previous slides. Once eligibility has been evaluated and the patient can and be enrolled in the study. It starts with the 3 steps that are typical for current CAR T treatment. The first step is being apheresis, the collection of the T cells. That apheresis product goes into production to CAR T cells, so to be transformed to CAR T cells. And during that process, 5 days before the administration of the final product, patients will undergo lymphodepletion. And then at day 0, they will receive the CAR T. And that phase is followed by a safety observation split in 2 parts, an immediate safety observation for those limiting toxicities and the immediate effects on cytokine release and or icons and other major events as explained by Tobias already in his example of the data. And then there is a longer term follow-up on efficacy and long-term safety. And there is an obligation to follow those patients to up to 15 years after the administration of the CAR T. And that has to be covered somehow in the setup of the study, or a separate observation study has to be designed. In practice of your clinic, it means a huge effort in collaboration between the different disciplines involved. Major players are the hematologists who do the apheresis, so the collection of the T cells. They mostly do the lymphodepletion as well, and they perform the administration of the CAR T cells. The screening is done by the rheumatologists in case of lupus. They are also monitoring the safety of the patient after the CAR T administration and the longer term follow-up. And they do the assessments for efficacy as well. There may be involvement of a nephrologist for the renal aspects, neurologists in case of observations of icons, the neurological side effects, and even dermatologists are sometimes involved to evaluate the skin. There are a few sites that have the option to produce CAR Ts themselves. And in that case, the pharmacy is also involved, and for the rescue of patients that experience severe side effects, the ICU is available, and pathology is sometimes needed to evaluate biopsies from the kidney or from the skin. That's a part of the assessments in the study. And then finally, the longer term follow-up on the last slide, thank you. This includes the 15 years follow-up as well, and monitored are the same assessments as in your study, but limited mostly to annual visits, but still include data collection of samples for pharmacology and immunogenicity, so antibodies against the drug, as well as safety and efficacy related information on the longer term and duration of the sustained efficacy or any relapse of the disease after a certain period, which is observed and described in literature as well as occurring in some of the patients. So I hope I have provided you with an informative overview of the background of study designing, and we are happy to collaborate with you if there's interest. Thank you for your attention and good luck with designing your own studies. And now I give back to Christina to guide us through the next parts of the presentation.
Thank you, Dirk, and thank you also, Tobias, because you really set very nicely an overview of what to be expected. This is not an easy endeavor to design these trials, and especially in autoimmunity, the diseases are so complex and the readout is so complex. So yeah, this was really important to consider. And now to add an extra layer of complication, I like to discuss about the manufacturing, because as you all know, cell therapy are a bit special. They are de facto considered drugs by the regulation, but they are a bit special drugs because of the complexity of manufacturing and also for the logistic of distribution. Majority of the CAR T in the field, the majority of cell therapy in the field are actually, as a commercial product, are autologous product. So we are going to focus on this for the next slides, but don't forget that actually cell therapy can also be manufactured from donor or from cell bank, for example, iPS-derived cells. In fact, this is a bit like the future, or like the dream of many many institutions and pharma to really be able to create a so-called off-the-shelf bank of therapies, so to remove the complexity of the autologous manufacturing. However, this is not yet completely proven to be efficacious, and a majority of therapy in the market, as I mentioned, are autologous. So autologous manufacturing requires about 7 to 12 days in the current manufacturing protocols just for the manufacturing step. And of course, a lot of more time for the logistic of the shipment of apheresis and final product. A cell product in CAR T, it is highly modified and manipulated. That's why it's considered a drug. Usually it is enriched. There is a cell culture to enable cells to be active and functional. And also, obviously, there is a gene modification, which also gives it another flavor or regulatory environment, which is the GMO regulation. Because as a drug, it needs to be monitored all over the manufacturing process with the so-called in-process controls. And also, there should be a proper quality control, especially for a commercial final product that has the QP and a strong responsibility from the sponsor, from the owner of the therapy, to guarantee safety via a proper quality control and quality management system. And then, as Dirk and Tobias already showed very nicely, that it's very important to monitor the patient, to measure efficacy, but most of all to check safety. And a final aspect is that cell therapy in autoimmunity, so CAR T in autoimmunity and immune reset, is extremely interesting from an immunological point of view, because you are kind of looking what happens in the patient once the immune system restarts from scratch. So there's a lot of study happening in the tissue and in the blood, the so-called concomitant research to understand really the mechanism behind this effect, the so-called bed-to-bench side. So now talking about manufacturing, the problem and the beauty, say, of cell therapy is that the product is the process. So if you control the process of manufacturing, you in fact control the accessibility to the cell therapy. And this is really the main discussion happening in the cell therapy field at the moment. Majority of cell therapy are brought to the market by pharma because, as mentioned, they are drugs. So they need to develop, and you need a big capital and investment to bring them to the market. However, as we're going to see in the next slides, many hospitals are also equipped to manufacture cell therapy, and they can also provide therapy in an early phase and in some special regulatory environment directly to the patient in the hospital. And finally, of course, manufacturing can also be done by third party, like private or for profit, but also non-profit organization that act like a cell development and manufacturing organization. Now let's have a quick look about the three possible ways to manufacture cell therapies. A pharma company manufactures cell therapy for two purposes, either for a commercial product or because it's still in an early phase and in a phase of development. So as a company-initiated trial. So now if you wish to, of course, bring your patient to one of these trials, of course, the company has to have a program in your country and approach your hospital you cannot recruit your patient in such a trial or eventually in future as a prescribing a product. This is, of course, the best way and the most proper way to really, make sure the product is reimbursed and everything is covered. However, of course, you need to imagine that there are, of course, parts in the world where these products are not available because the pharma are not reaching out those countries and those hospitals, and also big discussion right now in cell therapy is that if a central manufacturer by a pharma brings a long vein-to-vein time. So the patient might wait quite long before getting a cell. And is this, especially in cancer, is quite an important aspect, a bit less in autoimmunity, that some patients do not qualify for those trials or for those commercial products because they are too fast progressing. And also, finally, there is also another aspect that, of course, there's a low flexibility and not every patient of every indication qualify for those products. For that reason, a lot of hospitals in the world has actually moved to a so-called local manufacturing. So they equip themselves with a GMP facility, which actually usually was the current bone marrow transplantation unit. And they expanded to a cell manufacturing license to manufacture CAR T or other cell product. In this way, the so-called point-of-care approach allows early access, experimental therapy in first-in-human, early phase, and also, as I mentioned, to allow those patients fast progressing to get a quick therapy that they won't be able to get from a pharmaceutical product. And finally, also, they can do a lot of research because they really can control the product, study the product, and there's no this kind of black box effect that happened with a pharmaceutical product. The disadvantage of this approach is that, of course, a hospital needs to invest upfront a lot of money to set up a facility and to get a manufacturing license. And of course, they need to get reimbursed for this product because as an investigator-initiated trial, they are the sponsor, so they need to fund the funding. And finally, very importantly, cell therapy are drug. So it is actually not allowed by regulation for this hospital to treat patients in a routine way. They can only treat exceptional cases, as we saw for an example of the first patient treated in Erlangen or in other hospital for autoimmunity. The third aspect is the outsourcing. So if you are new in the field and you wish to establish a program for CAR-T treatment, you might also think to collaborate with a site that already have experience in manufacturing, and this could be like the third approach. So now let's have a look quickly into what is needed to manufacture your own product if you wish to address, let's say, the second case and start to treat some specific patient in a clinical trial, for example. So regulation beyond manufacturing and cell and gene therapy in general is extremely complex. And I don't want to go into detail here, but as you can see already in the European framework, there are so many aspects to consider, especially if one wishes to go to marketing authorization. So a pharma will have to look into all those bubbles, basically. But as a manufacturer in a hospital, the first basic information you need to consider are basically these four, and they are the most important is actually the GMP manufacturing license. So to build a solid base to really make sure that you manufacture a quality product that is approved for the manufacturing aspect, and then of course on top of that you will have to generate your clinical trial if you wish to investigator-initiated trial. And as we heard from Dirk, it's very complex process as well, but there are a lot of experience now in the field and people that can support you in such a design. And finally, I would like to touch in the last minutes about the reimbursement aspects. So let's start from the manufacturing. Building a cell and gene therapy product, so manufacturing cell and gene therapy product means that you need to have a GMP manufacturing condition because the cells are drugs. Now, typically a drug is sterilized at the end of manufacturing, but cells cannot be autoclaved because they would die. So the only way for, according to regulation, it is working in a complete clean environment all the time. Now, if you do this manually, you will have a lot of open steps. So you need to kind of build a mini closed environment for each manufacturing product, because in autologous manufacturing, every patient has a different separate product. So you need to keep those cells separated in time and space. This includes a lot of high cost. You cannot do parallel manufacturing in the same building, in the same room. You have a high cost of personnel training, higher risk of error mistakes, and also a high complexity in maintaining your facility and your instruments quality control. An alternative to that, a smart alternative to that, is to produce in a closed system. So according to the UDRLEX, it is possible when you work in a closed system to even work in a grade D. So you need to prove to your authorities that the system you're working with is closed and is sterile and produce a sterile product. And this is an example of how we approach also in our cell factory. This is a picture of our cell factory, this issue. So we use a closed system, the CleanMax Prodigy, and we've set up a so-called ballroom concept, which means that several instruments can be placed in the same room, and potentially you can outscale as much as you want. And also very important, we connect always and integrate our manufacturing with a proper quality control, because as mentioned before, it's important to also monitor your product overall, the manufacturing. So here's an example of our facility in Bergisch Gladbach, Germany. We have a middle-high scale, so manufacturing, because we are both CDMO and basically manufacture for our own product. And we produce about 600 products per year. So for that, we need 26 Prodigy, 4 Quant, Max Quant, and analyzer, and also about 17 people as personnel working there for 500 square meter space. This is, as I said, more like industrial scale, but I want to give you an example of what can be done in a smaller scale. So in a so-called point-of-care hospital manufacturing. This is a nice paper from the group in Saudi Arabia that established a CAR-T program and actually manufactures cell therapy in ISO 7 lab, so class CNC. And this is how they maintain actually the instruments you see on the bench, and not very fancy to be honest, and because they can manufacture in low scale, but they can still really go for a very effective and safe product for the patients, and they already treat quite a lot of patients in the region. So this was just an idea of what you can do for all manufacturing. And as I mentioned, you can also think about outsource your manufacturing. You can use CDMO services, of course, but they come with a cost, of course. Or you can also look for a partner in a hematology field that has already established a manufacturing site. And we can help you actually to find those partners, because our system, the Climax Prodigy, has been already placed in several institutions, hospitals in the world, as you can see from this overview from some selected publication. Really, literally in every site and continent in the globe, and we are happy also to support you and to build up this network, so you can build up your collaboration and get support for manufacturing eventually also from other hospital and sites. And so to conclude, there's a very uphill to manufacture cell therapies, but also to reach then eventually, if you wish, a marketing authorization or reimbursement, and of course before clinical trials. There are also alternative paths, which we don't have time to discuss now, but it could be the hospital exemption path, for example, in Europe, or other alternative way to treat patients. And the whole way up is, of course, very complex. So we have collected a lot of experience as Miltenyi Group, and we are really happy to support you. We provide, of course, tools and research product for the drug discovery and concomitant research. And as mentioned, a lot of manufacturing analytic platform, raw material, viral vectors, and services to support you to design a facility, to follow a protocol and blueprint if you want to have a manufacturing license for CAR-T. We have a big regulatory team that can support you also with consultation and also contract development services from our CDMO services. Just very quickly, I mean, don't have to go into detail here. This all is based on the concept that, as I mentioned, the Climax Prodigy is a closed system. So it is integrated, automated, closed, and also proven, as I show in many publications. And also we have a lot of proven data on our lentiviral vectors that have been now tested and used in many clinical trials in the world. It is off-the-shelf product, can be basically purchased, and it can be used, and also we can provide also the preclinical and clinical information related to it, and certify quality with a lot of documentation of our product. And finally, we also provide detection reagents and other monitoring tools to kind of track back your CAR-T in the patient after the treatments. And finally, as I mentioned, there are also CDMO services, so it's possible even to basically outsource completely to us the manufacturing of the cells, and this is just an example of how you can partner with others to get your cells manufactured. So now I would like to, for a few minutes, to close with some observation about the accessibility of cell therapies. It is clear that cell therapy has big potential, but there's a lot of geographical limitation. We know a majority of countries in the world don't have access to this therapy, because also pharmaceutical companies struggle to initiate a registration in every country. In the case of autoimmunity, I think we need to talk about rare diseases, Many autoimmune diseases are rare and ultra rare, so we can expect a pharmaceutical company might not be able to register product for those specific label. So you might want to consider point of care for treatment of exceptional cases that don't follow in the label of treatments of existing treatments. And finally, also we need to discuss the reimbursement system, because of course cell therapy have so these therapies have a big value, and there should be a policy discussion with the governments to make sure that eventually at one point these products are reimbursed. Just to give you a quick information about what can be discussed in terms of reimbursement, in the word of Georg Schettler and in this Nature Review paper, the pharmacoeconomic model can be designed to really balance what is the cost of standard of care versus the cost of CAR-T. CAR-T seems very expensive because they are one-time treatment of a few $100,000. But when you sum up all the costs of a standard of care and also of the different potential organ failure, hospitalization costs, and so on of a patient, you might get actually a positive balance. So this is type of discussion that can be done with authorities. And I invite you also to look up this other paper, a recent paper from the same group that really calculated not only standard of care cost, but also quality of life saving in treating a patient that can basically recover from a disease and go back to work. So this is also important aspect to consider. I would just summarize very quickly what we discussed today. There's still plenty of discussion we can do on immunological side, which are the right targets, how can you really move into allogeneic CAR-T. In terms of clinical trial, we have seen there are a lot of indication that can be treated. The discussion on lymphodepletion is still going on in terms of trial design, which type of monitoring is needed. I think we saw from Dirk a lot of insights for further discussion. And then I hope I gave you some insight on manufacturing. So it's an expensive process, but can be also reduced in terms of costs. There's a lot of discussion how to reduce and simplify manufacturing. And also, I hope I give you some insights on point of care and opportunities in local and distributed manufacturing. And finally, I really invite everybody to bring up to the public the discussion on reimbursement of cell therapies, because they are really promising, but at the moment they face this high cost. And I think governments and policymakers need to be aware about the potential of these therapies and also rediscuss the reimbursement system.
Thank you, Christina, and thank to all our speakers for sharing their valuable insights today. If you would like to keep the learning going, just scan the QR code to access additional resources, recorded talks, and a way to stay connected.